Alright, so now we're actually going to be talking about cell division. So meiosis consists of two stages, meiosis 1 and meiosis 2. So meiosis 1 begins with prophase 1. Now during prophase 1 or meiosis 1, something very special happens. So the homologous chromosomes are very close to one another. They're actually so close that they're physically touching each other. And everywhere they're touching, they begin to exchange DNA or genetic material. So this is the process that we call crossing over. So crossing over is when the homologous chromosomes are so close to one another that they're physically touching and they're actually exchanging DNA. So it means that you're going to have crossing over between chromosome 1 of mom and chromosome 1 of dad, chromosome 2 of mom and chromosome 2 of dad. You are not going to have crossing over between chromosome 1 and chromosome 2 or chromosome 1 and chromosome 3. Okay, that's something that we must remember. Crossing over happens only between homologous chromosomes. The other thing that happens during prophase 1 is that the meiotic spindle begins to form. So now the cell is going to go on to the next phase of meiosis, which is metaphase. So during metaphase, what happens is that homologous chromosomes align in the middle. They are no longer physically touching, they're just basically aligning in the middle. Now, if we take a look at this chromosome, originally this chromosome was all blue. And the other chromosome that is basically to the right, it was all pink. But because of crossing over that happened in the prophase 1, now you see that the blue chromosome now has a little bit of pink, and the pink chromosome now has a little bit of blue, signifying, if you will, crossing over. Now, during metaphase, the alignment of the chromosomes during metaphase, when the metaphase is going to determine what cell is going to end up with what chromosome. So, for example, in this particular example, that chromosome blue is on your left, and the chromosome pink is on the right. But it could have easily been the opposite, right? So the pink chromosome could be to your left, and the blue chromosome could have been to the right. So again, the alignment of the chromosome during metaphase is going to determine what cell is going to end out with what chromosome. All right, during metaphase, the meiotic spindle has fully formed, and they're actually attaching to each of those chromosomes. So now the cell is going to go on to the next phase. So anaphase 1 of meiosis 1. So what happens during anaphase is the meiotic spindle begins to become shorter and shorter, and it begins to pull those chromosomes to opposite ends of the cell. Once this happens, then the cell is going to go on to the next phase of meiosis. In this case, we're talking about telophase and cytokinesis. So what happens during telophase and cytokinesis is that the chromosomes have reached opposite ends of the cell, and the cytoplasm begins to split in half. Alright, so now we're going to start meiosis 2. So meiosis 2 begins with prophase 2. So what happens during prophase 2 is that the meiotic spindle begins to form. So once this happens, then the cell is going to go on to the next phase of meiosis 2. In this case, we're talking about metaphase 2 of meiosis 2. So what happens with a metaphase is that the chromosome align in the middle, and the meiotic spindle actually attaches to each of those chromosomes. So once this happens, then the cell is going to go on to the next phase. In this case, we're talking about anaphase 2 of meiosis 2. So what happens with anaphase is that the meiotic spindle becomes shorter and shorter, and they're actually separating those chromosomes. It is extremely important for this process to happen because having too many chromosomes is not a good thing, and having too little chromosomes is not a good thing. So sometimes there are some mistakes that actually happens where the chromosomes fail to separate, and what happens is that a cell is going to end up with too many chromosomes. A typical example of this is Down syndrome. Alright, so now we're going to go on to the next phase of meiosis 2. So telophase 2 in cytokinesis. So what happens during telophase 2 in cytokinesis is that the chromosomes have reached opposite ends of the cell and the cytoplasm is split in two. So that at the end of meiosis, we have made 4 cells. And what's important about these cells is that they have half the number of chromosomes. And when you look at these chromosomes, they look different than the chromosomes that you find on the parent cell. So one of the things that I tell my students is that meiosis is important for diversity. And diversity is good. So just to summarize, so at the end of meiosis, we have created 4 cells. And each of those cells have half the number of chromosomes than the parent cell. And when you look at each of those chromosomes, they look different than the chromosomes that are found on the parent cell. So each of these cells represents either egg or sperms. So now you know, meiosis is important for creating those gametes. Meiosis is important for creating diversity. Alright, so let's go back to our story of cystic fibrosis. So even though individuals who are what we call normal, right, which means that they don't have cystic fibrosis, they can still have an offspring with cystic fibrosis. And the question is, why? If those individuals express a normal phenotype, which means that they don't have cystic fibrosis, how is it possible that they can still have an offspring with cystic fibrosis? So let's find out how is this possible. Alright, so cystic fibrosis is caused by recessive alleles. So these are 2 terms that you must know. So recessive versus dominant. So what do we mean by dominant alleles? So it basically means that a dominant allele is going to be more phenotypically expressed in a population. When it comes to a recessive allele, it's only going to be observed when you actually have 2 copies of the recessive allele. Okay? So a good way to think about it is basically when it comes to the dominant alleles, is that they're producing more of the protein. So in science, we basically designed a lowercase letters to represent a recessive allele, and we use uppercase letters to designate a dominant allele. So in this particular example, we're using uppercase letter A to represent a dominant allele and lowercase a to represent the recessive allele. In the case of cystic fibrosis, only individuals who have 2 copies of the recessive allele are the ones that are going to express cystic fibrosis. Alright, so here's some more terminology that you must know. So a heterozygote or heterozygous. So this is individual who have 2 different copies of the allele. So in this case, we're talking about a dominant allele and a recessive allele. Now, in this particular case, those individuals are going to have the normal phenotype, which means that they're not going to have cystic fibrosis. However, because they have 1 recessive allele, they can actually pass that recessive allele to an offspring. So another term that we use for heterozygous is actually carriers. Now you have what is called a homozygote, so which means that you have 2 identical copies of the allele. So you can have homozygous dominant, which means that you have 2 copies of the dominant allele, so in this case 2 uppercase A's. So these individuals are going to be phenotypically normal, and they would only pass the dominant allele to their offsprings. Now you can also have what is called homozygous recessive, which means that these are the individuals, in the case of cystic fibrosis, those are the individuals that actually have cystic fibrosis because they have 2 copies of the recessive allele. Again, heterozygous, 2 different alleles, big A and lowercase a. Homozygote, you can have homozygous dominant, which means 2 dominant alleles, or you can be homozygous recessive, which means that you have two of the recessive alleles, and those are the individuals that actually have cystic fibrosis.